So this is Population Genetics, Chapter 23, and this is pages 44 to 502 in your text. And we'll be talking about a little bit more in depth about genetics, and we'll be talking about some equations that we use to estimate how genetics moves and how genetics influences populations. And so this will probably be the most math-heavy chapter that we'll cover this entire semester. Okay, and it'll be this one in chapter 22 that will be on your exam well, depends on when you're watching this, right? Thursday, this coming Thursday. Okay, so let's get started. Now, there are basically two types of evolution that we'll be talking about. There's going to be microevolution and macroevolution. All right, so microevolution is basically evolution within specific species. And this is what this chapter is all about. Okay, we're talking about allele frequency and how allele frequencies change over time depending on what happens to the population. Okay, the other one is macroevolution, and generally a buildup of microevolutionary changes can often lead to a macroevolutionary change, something like a species changing and not being able to mate with another population, for example. And so microevolutionary changes lead up to a macroevolutionary change. We won't be talking about macroevolution in this chapter. We'll be leaving that for in the coming weeks. Okay, so let's talk a little bit about allele frequency. What is allele frequency? What does it mean? How do we calculate it? And how do we calculate its changes in estimating microevolution? So first, before we do that, we need to talk about what a population is. Now, a population is basically going to be a group of organisms within a specific area. Here I'm asking, what is the difference between a population and a community? Well, I just defined a population. A community is multiple different species within an area, including the abiotic factors such as the water, the air, everything like that. And so a community takes into account many species. A population is many organisms of a single species. Okay, that's the big difference. Now, another term here we need to go over is a gene pool. A gene pool is basically all of the organisms' genetics and genes within that population. Okay? And so the gene pool is what creates genetic variation. Basically, the amount of genetic differences among organisms within that population. So a little bit about genetic variation. And I know it seems like this PowerPoint isn't showing up very well. So if you can't see any of these images, make sure you go to ACE, download the PowerPoint, and you can see them a lot clearer. Okay, so phenotype variation is basically going to be one of the ways that genetic variation is reflected. So we talked about a gene pool, we talked about genetic variation. A gene pool is basically all the differences within that gene pool are going to be based on two things. Phenotypic variation, okay, which is here, and environmental variation. So basically, how the organism looks is going to be due to one of those two things. Is it because of how the genes affect the organism, right? How the genetics affect the organism. So we talked a little bit about this in the last couple of weeks, right? You can have good genes, but if you don't eat well, you're not going to have the right phenotype. So the same thing here. And so genes can affect how the phenotype of the organism looks. Basically, is it tall? Is it short? Is it green? Is it red? Your genes are going to basically result in whatever you look like. But it can also be due to the environment, which is what we talked about before, right? And so the phenotypic variation, in other words, the amount of differences in a population, like how they all look different, it can be due to what the organisms are eating, what type of environment they're in, things like that, climate, habitat, All those things can influence how the organisms look too, right? And so here are some examples on here. So this is the same species of caterpillar on the left. Its entire life it's been eating the flowers of the oak tree. What happens is certain genes get turned on, and because of that, it looks a lot like oak flowers, camouflage. On the right, same thing. All it eats is oak leaves, but the difference is it looks more like a stem or a branch. So certain genes get turned on, turned off, and then its phenotype reflects that. So this variation that we talked about within populations, right, either due to genes or due to an interaction between the environment and the genes, where does it come from? Well, there are several different places where it comes from. One of which, so there are four sources. The one of which here are new alleles. So basically mutations can arise, pop up, and then influence how that species looks, how that organism looks, and then if that's a good mutation, then it can spread throughout the population and eventually become a dominant allele within the population. Also, gene number and position can be altered based on things like crossing over, right? We talked about that. And so the position of where that gene is, maybe it's on 1M, and then crossing over occurs, now it's on 1P. So things like this genetic recombination, that can also affect where the gene is, and it can affect genetic variation and how much there is in the population. Also, the sole act of reproducing quickly can affect genetic variation. The way this occurs is that, remember, we talked about genetic recombination. Sexual reproduction depends upon genetic recombination to change the next generation. So if a species is reproducing quickly, then it has a lot of genetic recombination. Every single time it reproduces and makes a new generation, genes get mixed, genes get mixed, gene gets mixed over and over and over. So the more quickly it reproduces, the more genetic recombination there is. Okay, and lastly, sexual reproduction in general versus an asexual species. Sexual species are going to have more genetic recombination, more genetic variation in their populations than asexual species will. Okay, so let's talk about that allele frequency in microevolution that we mentioned. And so what microevolution really is, and we defined this already, but I'll define it again, is a shift or a change in allele frequency over time. That's all microevolution is. So what is allele frequency? Well, it's pretty easy to define. The frequency of particular alleles in a population. All right? So there you go. The proportion of a particular allele of a gene in a population. That's what allele frequency is. What does that mean? Well, let's say this is our population here, okay, for this particular gene. I don't know if you can see that or not, but we have big R are the red flowers, big R are the pink flowers, little r are the white flowers. Okay, we've been dealing with things like this in Mendelian genetics before. Okay, so if this is our example here, let's say we have 500 of these flowers. All right, and of those 500, 320 of them are homozygous dominant, so red. 160 are pink or heterozygous, and 20 are homozygous recessive. How do we calculate allele frequency with these numbers? So notice what I've given you are the genotype, basically the numbers of each genotype in the population, right? 320, 160, and 20. For every genotype, I gave you the numbers. If you wanted to, for example, figure out the genotype frequency, all I need to do, for big R, divide 320 by 500, which is the total, and you have the genotype frequency for big R. You want the genotype frequency of big R, divide 160 by 500, voila, you have the genotype frequency for the heterozygote. And the same thing for the homozygote, divide the 20 by 500, and you have the genotype frequency of the little r, right? But we want the allele frequency. So how do we get the allele frequency? So for example, one of the alleles here is big R. The other allele here is little r. So what if we want to know in this population, what is the frequency of big R? Or what is their frequency of little r? How do we do that? Well, so as I said, you have 320 big R, 160 big R, little r, and 20 little r, little r. The frequency of the big R allele, remember those genotype frequencies I was just telling you how to calculate? Here's the equation for figuring out allele frequency by using those genotype frequencies. So frequency of the big R allele, you take the genotype frequency of big R, so that would be 320 divided by 500, and you add that to half of the heterozygote frequency. So half times 160 over 500, and that will give you the allele frequency for big R. Why half? Because notice that for homozygous dominant, you have two big R's. So we're interested in the entire thing because we're interested in big R and the entire genotype has big R. But in this genotype, only half of the alleles are big R. So we're only interested in half of the alleles, so we multiply by half. Make sense? So likewise, the frequency of the little r allele is calculated by the little r genotype frequency, so 20 divided by 500, plus 1/2 of the heterozygote genotype frequency, 160 divided by 500, okay, times a half. So therefore, here we do the math, 320 divided by 500 plus 1/2 of 160 over 500 gives us 320 divided by 500 plus 80 divided by 500, right? Which equals 400 divided by 500. So the R frequency equals 80% or 0.8. Okay, so if you think about it, if the frequency of big R in the population is 0.8, what must be the frequency of little r? There's only two alleles for this gene, big R and little r. So if big R is 80%, little r must be 20%. Okay? Make sense? Why am I asking you that? I don't know. You can't answer me. Okay, so how do we know if this frequency changes at all, right? Let's say this is like a shot in time, right? 80%, 20%. But how do we know if it changes? Well, what we can do is we can compare this frequency to a null model, or basically, if this population never ever changes or is ideal, it should have these values. Okay, that's what a null model is. And so that's where we come into something called equilibrium population. Okay, an equilibrium population is an impossible one, but it's one that we use as a model for a population that never changes, never goes through evolution, never has any migration, never has any mutation, nothing ever happens to it, and it just stays the same forever. Okay, obviously that doesn't exist in real life, that's impossible in real life, but as I said, it's a null model. So what conditions does this equilibrium population have to meet? Well, I just said some of them, but here they are. Here's a list. So no mutation, right? No migration. The population has to be extremely large, and we'll talk about why in a second. There has to be random mating. Okay, we can't have mates choosing particular genotypes or phenotypes because that would influence how the population shifts. And there has to be no natural selection, right? Does this exist in nature? As I said, no, it does not. This is just a null model. An equilibrium population is not really true. It's a mathematical model. So it's basically used to calculate the expected ratios. So for example, allele frequency, genotype frequency compared to each other, like big R versus little r allele frequency, big R genotype frequency versus big R genotype frequency versus little r genotype frequency. It's used to compare them if no microevolution is occurring. And then once we have those numbers, we can compare them to the real life numbers and say, are they different? If they're different, we know evolution has occurred. All right, that's generally how it's used. So here's your example of a equilibrium population. Okay, I don't think you can see these numbers, but we have 490 big A butterflies, 420 big A butterflies, and then 90 little a butterflies in generation 1. Generation 2, we have the exact same numbers. Generation 3, we have the exact same numbers. 4, 5, 6, etc. The exact same numbers. Even though they're reproducing and reproducing, every generation has the exact same numbers. Impossible, right? It'll never happen. But that's what an equilibrium population is. So this is where we come into using something called the Hardy-Weinberg formula. And the Hardy-Weinberg formula is used to determine the ratios in an equilibrium population. So here is the equation for Hardy-Weinberg. Okay, it's p squared plus 2pq plus q squared equals 1. Okay, that's the more complicated formula. And then there's a second formula, which technically we've already used, but here it is again, p plus q equals 1. So what does p stand for? What does q stand for? What do these variables mean? Well, p equals the frequency of the dominant allele. So basically, in our previous example, this would be big R. Remember, this is very important. Remember, alleles are one letter. Big R, or say little r, those are each alleles. Genotypes are 2 letters, right? Or 4 letters, or 6 letters, or whatever. Basically, it's the entire genotype of the organism. So for a monohybrid cross, a genotype would be big R, little r, or big R, right? In a dihybrid cross, a genotype would be big A, big B, little b. That's a genotype. The alleles are big A, or little a, or big B, or little b, so every single letter is an allele. Okay, that's an important point. So P is the frequency of the dominant allele, so big R. Q is the frequency of the recessive allele. Okay, so therefore P squared is the frequency of big R, so basically the homozygote dominant genotype. 2pq is the frequency of the heterozygote genotype, and q squared is the frequency of the homozygous recessive genotype. Okay, so little r. So how do we use these formulas? So let's try to use p plus q equals 1. Technically, we already used p plus q equals 1 when I asked you that 80%, 20% question a few minutes ago. That was using p plus q equals 1. But let's see what this says. So if our population has 60% of it is q, 60% is q, what percent is 60% is big Q, what percent is little q? It has to be 40%. P equals 0.6. So 0.6 plus q equals 1. Move the 0.6 over. 1 minus 0.6 equals q, 0.4. Right? So that's using p plus q equals 1. Very simple. How do we use p squared plus 2pq plus q squared equals 1? Well, let's say this is our population again. We have 3 different flower genotypes. Big Q stands for are red flowers. Big Q, little q are also red flowers, so simple dominance. And little q are white flowers. So let's say a population has 36% red plants. What percentage of that population, of that 36%, are big Q, little q? Well, in these situations, we need to ask, what is it giving us? So it says 36% are red. and it wants what percentage is big Q little q. So in Hardy-Weinberg, what is big in the Hardy-Weinberg equation? What is big Q little q? It's represented by what variable? And the answer is 2pq. We just said that, right? Big Q little q in the Hardy-Weinberg equation. It's the heterozygote. The proportion of the heterozygote is 2pq. Okay. What are we given in the question? We're given 36% are red, right? What genotypes does 36% consist of then? Notice big Q is red, big Q and little q are red. So 36% equals the frequency of the big Q, and the big Q, little q. So this is unusable to us because 36% is equal to the frequency of big Q, which we know is p squared, plus the frequency of big Q, little q, which, as we just said, is 2pq. So we have two variables there, p and q equals 36%, right? And so we can't solve for that. And so what we do instead is we think of our third genotype, little q, right? The white flowers. So if 36% of our population are red, what percentage of our population is white? Well, it has to be 64%. So what's white? White is little q. So in these equations, you're almost always going to try to get the homozygous recessive genotype frequency first, okay, and then get the recessive allele second, okay? So 64% has to be little q because we're given 36% red, so this has to be 64%. Okay, so 0.64 equals the proportion of our homozygous recessive. In Hardy-Weinberg, what is our homozygous recessive represented by? It's represented by q squared. So q squared equals 0.64. Okay, so if q squared equals 0.64, how do we get q? If you remember from algebra class, you basically square root of 0.64 will give you q. Okay, so q is going to equal 0.8. That's something we can work with. Remember, our equation p plus q equals 1. Well, if q is 0.8, p has to be 0.2. So now we have p, and now we have q. We can easily solve for what it's asking for, which is 2pq. So 2 times p, which is 0.2, times q, which is 0.8. Voila, we get our answer, right? P is 0.2. What is q squared? So our frequency of q squared should end up being 0.32 or 32%. Okay, this is the most complicated of all the equations we'll be doing, of all the problems we'll be doing. Okay, so we'll do some practice on this this week and next week. So for Hardy-Weinberg, we said it has to have 5 conditions that it has to meet, right? It has to meet no mutation, random mating, the population has to be very large, there has to be no migration, there has to be no natural selection. Of those, some are minor violations where if they're violated, the allele frequencies don't shift that much really. And then some are major violations where if they're violated, then the entire population shifts and is definitely not in equilibrium population. So let's discuss which are which. So the minor violations include having non-random mating instead of random mating. So this changes genotype frequencies, but it doesn't shift allele frequencies very much. Also mutations. Mutations are rather rare, and so because they're rather rare, having mutations in a population is a minor violation. But some of these are major violations of Hardy-Weinberg, and so they bring about major microevolutionary change. So let's talk about those three that are major violations of Hardy-Weinberg. These include, number one, having a small population. If you have a small population, something called genetic drift will occur. What is genetic drift? Well, genetic drift is basically microevolution from random fluctuations in the population. So for example, if you have a population of 1 million spiders, okay, let's say some type of disease goes through that population of 1 million spiders and five of them die off. 5 of them dying in a 1 million spider population is going to be a very minor blip, right, on the history of the evolution of that population. But let's say your population was 10 spiders and then 5 die off. That's half of your population dying. So that's going to be a major change. Okay, so having a small population can really have genetic drift, which is just random fluctuations due to all kinds of different genetic things, can really have an effect if it's a small population. And genetic drift, by definition, is random, right? It can be disasters occurring. It can be, like I said, like some disease knocking some population back. It can be anything just random that affects the population. That's genetic drift. And so genetic drift, by definition, reduces variability in the population because it's going to kill off some of the members of the population. It also usually reduces something called heterozygosity. Heterozygosity is a big word that just means the proportion of heterozygotes in the population. So usually genetic drift reduces the genetic variation in the population and reduces the amount of heterozygotes in the population. And it can lead to something called fixation and loss. Okay, fixation is where an allele, say like big R, goes all the way up to 100% of the population. So every single individual in that population is big R, big R. And so therefore the allele frequency of big R is 100%. And so therefore, remember your P plus Q equals 1. If big R is 1, little r has to be 0. Okay, so little r goes to 0, which is called loss. You have completely lost little r from the population. It's impossible to get it back unless you have some migrants come in who have little r, or a mutation evolves to get little r back. Other than that, I mean, you lost little r. So that's called loss when it goes to 0, and fixation when it goes to 1 or 100%. And so that can happen from genetic drift as well. Let me show you an example. So if your population is very, very large, so this middle blue line here, random fluctuations that happen, some organisms die off, some random accidents, or just diseases or disasters or whatever, small fluctuations in the population are from genetic drift. And so your population is very large, like this blue one is here, the fluctuations are kind of small. Because like I said, 5 out of 1 million spiders, not a big deal. But if your population is small, like this red one or this red one, where your population in this example is 10, in your genetic variation, those random losses of individuals really has a big effect on your population, because you're losing one individual, that's 10% of your population gone. And so those random effects can bring it, for example here, your big B went to fixation and little b was lost. And in this case, the opposite happened. little b went to fixation, and big B was lost. Again, that's just random. It just randomly happened. So a special type of genetic drift is called founder effect. And this is when a small subset of a population leaves and founds a new population on a different location. So that newly founded population took a random subset of the population, the original population, and founded your new location. So because it was a random subset, it just happened that in this case we didn't get any yellow alleles. Excuse me. So because of that, your new population has 0 yellow. So again, a random occurrence affects the genotype of the population. Same thing for another type of genetic drift called population bottleneck, where you have your population that's very diverse, genetically diverse on the left, you have yellow and red beetles, and then a disaster strikes, it reduces the population all the way down to 2 beetles, and all the yellows die, and all you're left with are the 2 reds. And then once they try to rebuild this population, they have to rebuild it from the 2 reds, and so your new population, it has no yellows. Okay, so this just genetic randomness, this disaster, killed off a lot of your genetic variability, okay, and one of your phenotypes was lost. That's genetic drift. So this is one of the 3 violations that are major violations of Hardy-Weinberg. Okay, another one is gene flow. Gene flow is another term for migration. Okay, but basically transfer of alleles among population. It doesn't necessarily have to be individuals or organisms. It could be seeds flying on the wind. It could be a coconut floating in the water and moving across the ocean, right? That organism is migrating to a new location. It's not an organism yet, right? It's just a seed, but that is gene flow. And so, again, this is random, right? We don't know who's going where at any time. We don't know where that coconut's floating. We don't know where the seeds are flying. All this is random, just like genetic drift is random. Gene flow in general reduces the differences between populations over time, right? If you think about 2 populations and they're not mixing, if they're not mixing, they're going to evolve on their own paths and become different and different and different and different as their environments affect them and they adapt to their individual environments. But as you get more mixing between these 2 populations, they start to become more similar and more similar and more similar as the alleles start to mix together. Okay, so usually gene flow reduces differences between populations. Our third violation that has major impacts on Hardy-Weinberg is natural selection. Okay, now if we look back at all those types of violations of Hardy-Weinberg, remember the 5? We're going to see, do they affect allele frequency randomly or non-randomly? Well, genetic drift, like we just said, is random. Gene flow, like we just said, is random. Mutations are, by definition, random. The only 2 that are non-random are non-random mating, which we said before is a minor violation, so not that big of a deal, and natural selection. So natural selection is the only violation of Hardy-Weinberg that is non-random and it has major effects on the allele and genotype frequency changes. Okay, it's an important point. I may ask you that on a quiz or an exam. Right. So what is natural selection? How does it work? Natural selection largely depends on something called selection pressure. Okay, selective pressure. Selective pressure is basically any factor that causes disproportionate survival and or disproportionate reproduction, right? So a selective pressure is something that causes a species to survive longer or have twice as many babies And so a selective pressure will influence those things and cause it to have more babies or die quicker or live longer. Okay, so there's two main kinds of selective pressures. One is an environmental, basically called non-living environmental selective pressure. So these are things like how hot it is, the temperature, weather, how many disasters there are, how much shelter there is in an environment. That can affect who lives, who dies, right? Think of like an Arctic hare, like a bunny covered in white fur. You know, this bunny depends upon Arctic habitats with snow, cold temperatures, and a frigid environment. You put that Arctic bunny in the Sahara Desert, there's no way it's going to survive, right? And so this is the selective pressure would be the environment affecting that species, okay, affecting its survivability and affecting its ability to reproduce. The other is, of course, the living environmental selective pressure, so other animals or plants or whatever organisms affecting it, right? So there's three types of living environmental selective pressures. The first of which, like shown here, is competition. So competition is always between members of, doesn't have to be members of the same species, but two or more organisms of either the same or different species interacting and both negatively affecting each other, right? So we call that antagonistic. In other words, species A would be able to eat a lot of food if species B wasn't there. Same thing for species B. They would be able to eat a lot of food if species A wasn't there. But because they're both there competing, they both eat less food. Okay, you'll learn more about this in ecology when you take that. Another type of selective pressure is predation, right? Predator-prey interactions. This is a positive-negative interaction where it's great for the predator, right? Very positive for the predator, not so great for the prey, okay? And so it's where one organism is preying on another. Obviously, this is an extremely strong selective pressure, right? Because where in competition, yeah, you may not eat as much and you may have to spend some resources to compete. You're not dying because of it, okay? Well, not always. Usually you're not dying because of it. But in predator-prey, you're dying. You're getting eaten. And so all your alleles, all your genotypes are gone if you get preyed upon and eaten. So predator-prey interaction is an extremely strong, probably the strongest selective pressure. Basically, the pressure is don't die. So this is where camouflage comes from. This is where warning coloration comes from. This is where being fast or being able to hide, all those things are largely due to predator-prey interactions. Extremely strong selective pressure. And the last one is sexual selection. So being able to mate more or less, depending on how successful you are. And so this can be either intrasexual selection or intersexual selection. Intrasexual selection is between the, so basically selective competition, sexual competition between members of the same sex. So as you see here, like you have the elk, the male elk, basically battling each other for control over the female herd. And that's intra. And intersexual selection is often called mate choice, where the female gets to select which male she wants to mate with. So think like peacocks, right? So the male peacock looks really pretty, has like the pretty display, basically showing off. So hopefully he'll get chosen by the female so that he can pass on the genes. And she's looking for somebody that has good genes and is healthy and somebody who can help give her healthy offspring. Okay, so that's intersexual selection. So how do these frequencies change and shift over time as the populations evolve? Well, there's So natural selection basically causes populations to shift in three different ways, three different modes. Okay, and so if this is your population here, you have your bell curve and you have phenotypes on the x-axis. In this case, you have different colors of mice. You have dark mice on the right, brown mice in the middle, and white mice on the left. And then the frequency is on the y-axis, with the highest frequency being there. So one of the modes of natural selection is called directional selection. In other words, one of these extreme phenotypes on the right or left is the selective pressures basically pressure it to the right in this case. And so these darker mice survive at greater rates, reproduce at greater rates. These mice on the left survive at lower rates, reproduce at lower rates. And so your population shifts to the right. Okay? One of the examples that can cause this is called environmental change. And so, in other words, the environment shifts. Say you have a brown sandy habitat initially, but then it shifts to more lighter or whiter sand. If that happens, the white mice may blend in more and survive more and have more babies because they're not eaten as much. And then the dark mice may die off because they're eaten more and captured more. A second type of a mode of natural selection is called disruptive selection. And disruptive selection is basically where the phenotype frequency shifts for both extremes. And so both extremes, both the right and the left in this case, are selected for, and the middle is selected against. Okay, this is often a response to two extreme environments or two different types of survival strategies. And so you have a habitat with sand and rocks, and you have one species blending into the light colored sand, and then another species blending into the dark rocks. But if you're in between color, you don't blend into either one. And so you get the one that's not blending into either one gets eaten more, less camouflage, dies off. The other two are camouflaged and survive. And then our last one is called stabilizing selection. And stabilizing selection is the opposite of disruptive, where instead of your two extremes are selected for, your two extremes are selected against, and your mid phenotype is selected for, and it goes up in frequency, and the others go down. Generally, this reduces genetic variation in a population because you're losing the tails. Notice you're losing that tail and that tail. And an example of this is human baby birth weight. Heavier babies are more likely to die if they're overweight when they're born. And then, of course, underweight babies are also more likely to die if they're born underweight. But babies that are in a middle weight range usually survive at a higher rate. So we talk about all these phenotypes. How do these phenotypes come along? How do new phenotypes arise in a population? Well, generally, new alleles usually arise by mutation. If they arise by mutation, they can become new phenotypes. If those new phenotypes allow the organism to survive and reproduce at a greater rate, they're called adaptations. Okay? So do all new phenotypes that arise from mutation, are all those new phenotypes adaptations? No. If a new phenotype arises, that gives somebody 6 fingers instead of 5 fingers, and it doesn't help them survive or reproduce at a greater rate, that's not an adaptation. Okay? That's just a new phenotype. Okay? An adaptation, by definition, has to increase your ability to survive and reproduce. Okay, we call that increased fitness. Okay, so I gave a little flow chart here. Does it increase your relative fitness? So let's talk about the word fitness and relative fitness and what that means. Fitness is basically the amount that you reproduce in life. Okay, your biological measure of your success in life. How many kids do you have? Right? I have 0 kids, So my fitness is 0. I am not successful at all. Because I have a cat, that does not count. Does not count. Okay, so it doesn't count. So my fitness is 0. All right, and so this is the amount of DNA you contribute to the gene pool. That is your fitness in life. Relative fitness is related. It's the fitness that you and your direct relatives contribute to the next generation. Okay, so for example, I have a brother. My brother has kids, and so that increases my relative fitness because he shares some of my alleles, right? By definition, since he's my brother, he shares 50% of my alleles. But so therefore, he passed on 50% of his alleles, and so some of my same alleles are in my cousin, or sorry, nephew, right? My nephew. So that's relative fitness, the amount of DNA that you and your direct relatives pass down to the next generation. So fitness is what you're passing down. Relative fitness is what you and your direct relatives are passing down. And so looking at adaptation, a phenotype is an adaptation if that adaptation increases your relative fitness. If it increases that organism's ability to either it passing down its genes at a greater rate or alleles at a greater rate, or its relatives being able to pass down the alleles at a greater rate. All right. So last thing we'll talk about is a few special types of natural selection. This is called balancing selection. Balancing selection is where natural selection preserves variation within a population, usually maintaining multiple forms. Okay, and so there's two kinds of balancing selection. One is called heterozygote advantage, and this is where, just like it sounds, the heterozygote has a higher relative fitness than either of the homozygotes, homozygote dominant or homozygote recessive. One of the examples of this is, again, sickle cell disease. And in sickle cell disease, like we talked about, there are some disadvantages to this, and we talked about pleiotropy and sickle cell. But individuals that are homozygous recessive for sickle cell, in other words, they have the full-blown sickle cell disease, have immunity to malaria, but because they are full-blown sickle cell, they usually have a lot of health problems and survive at lower rates. Those individuals that are homozygote dominant don't have sickle cell at all, but are prone to malaria and oftentimes die from malaria at higher rates. Those individuals that are heterozygous, sickle cell goes through partial dominance, so they have some cells that are sickled and some that are normal. So they do have some slight health issues, but they have some malaria resistance as well. And so these heterozygotes have a higher relative fitness than either the homozygotes in environments where malaria rates are very high. Okay, so that's heterozygote advantage. The last one I think is really awesome, and it's called frequency-dependent selection. And this basically says that the fitness of a particular phenotype is directly dependent upon how common or uncommon it is in the environment. Okay, let me give you an example. There's this fish, and this fish is the prey of another species of fish. The second species of fish is basically a scale eater. It eats the scales off of this host fish. And what it'll do is it'll swim up on the left side and then take a bite out of the fish and then swim away. Or it'll swim up on the right side and take a bite out of the fish and swim away. It doesn't want to get caught. If it gets caught by the host fish, it can die. And so some of these fish are born to bite on the left side, and some of these fish are born to bite on the right side. There's basically two different genotypes and therefore two different phenotypes within this population, bite on the left or bite on the right. And so you could imagine this host fish trying to catch these parasites and trying to prevent parasites from biting it. If your population of your parasites was almost 100% right side biters, then that fish would constantly be looking to the right to catch it. And then the left side biter would have an advantage because he's never looking its way. And then its frequency would go up. But as its frequency goes up over time, that host fish will start looking to the left now. And now the right's frequency has an advantage. So basically the right and the left go up and down in frequency as their how common they are goes up and down. Okay, that's a really cool example of how frequency dependent selection occurs. All right, and I think that's it. Yeah, scale-eating fish is that example. All right, so this ends that.